It will not be all boring lecture, it will be a very interactive class. So, the first question that I would like to ask all of you is, why did you register for this class? So, what I basically need from all of you, what's your name, where are you from, and maybe in a single sentence or two explain or let us know why are you interested about this class or in this area in general. And it could be anything, like you might want to say, like, you are just interested about photosynthetic microbes. It could be as simple as that. But I would like to go through all of you and need to know about your background. So, let's start from the back. Could you start?
I am a PhD scholar in IIT Kharagpur, School of Energy Science and Engineering. I registered in this course because I am interested about the evolution about the evolution how cyanobacteria evolve with the evolving atmosphere in the earth. That is why I am registered in this.
Good. Next. What are the backgrounds? Why you have taken this class? So, I could see three different themes. Exploratory theme, meaning that some of you would like to explore, would like to know about the field. Some of you work either in cyano, microalgae, some related field. Some of you work in a different system, but kind of want to learn some of the techniques. So, these are the three different themes that I could see. If you haven't seen already, I have sent an invitation to all of you to register for a coursework management system, which is called Canvas. So, the idea is to get your feedback on your learning so far. Why am I doing this?
If did not get the invite, you can ask.
Or have you not? Is there anyone who didn't get invite?
You can write the email to Bharat.
Okay. So, I think I got the list of the students and I sent an invite to all of you. So, basically there is a survey which I would like you to participate in to get a better feedback about your background. So, tomorrow and the day after I will do 3 different classes, but differently. I will talk about it later on. So, there is a reason why I am doing all this, and possibly I can talk about it later, because I think I have taken a lot of time. But anyway, so as I said, this is the first lecture on bioproduction. So, the way this lecture will go on is, I will pause once in a while, I will stop, I will ask question, and I will definitely need answer from you. If there are volunteers, it is good, otherwise I will point a specific person, since I do not know your names, and I do not think within 4 days I can know all of your names. So, let us do this, and whenever you have a question, please stop me. I mean, as I said, this class has to be interactive. All right. So, if we think about bioproduction, obviously you could see that this is probably related to a production of a biomolecules. When we talk about biomolecules, that means that definitely there is a biological system that is involved, right. So, most of the times here I am giving a very crude example where you can use sugar to produce succinic acid, which is a commercially used solvent for many different applications. And if you look in literature, you will see so many different systems can be used to produce succinic acid. The question is, some of the biological system can inherently produce a product of interest, some of them cannot. So, why are we interested in engineering a microbe? Can anyone give me an answer? Why engineering is needed?
So, to get to increase the production rate and to get desired products of the desired quality.
So, you basically mentioned about 2 different aspects. Desired product. In many ways, in many different systems, your inherent system cannot produce your desired product. The system might be efficient to utilize a specific nutrient. For instance, since this is a photosynthetic microbe based class, let us say we know a microorganism, a photosynthetic microbe that can use carbon dioxide and sunlight efficiently, but cannot produce your product of interest. Then it is your job to design that microbe so that you can get the product. Now, you can get the product, but you may not get it in a certain level. So, that is a different challenge. So, 2 different challenges. What is your desired product? Can the microbe produce the product? Can the microbe produce it in the desired quantity, right? Can we all agree to that? All right. So, let us think about this pathway. So, sugar is producing pyruvate, acetyl-CoA, and then through TCA cycle we can produce succinic acid. For the time being, let us assume that you know about this pathway. Many of you worked in metabolic engineering, many of you know about synthetic biology. So, let us say this is the enzyme E1 or the gene associated with this. What I am asking you, let us say I would like to upregulate the gene, or I would like to make sure the protein turnover for E1 is better than your previous case. And you could assume that the flow through this path would be higher, right, hypothetically. What I am asking, do not consider about all these other reactions, just think about this highlighted path. Can you see why that may not work? If I upregulate, if I change the protein content of E1 to produce the level of succinic acid, what could go wrong? Why it may not work? Or if you think it should work, back your answer. Why do you think it may or may not work? Like hypothetically, if this is a linear pathway, if you are upregulating a gene, you can assume that you will get more product, right? What I am asking, what could go wrong? Anyone who works in metabolic engineering should know this. Who is working in metabolic engineering? Okay, for this time being, let me give you the answer. So, now when you are thinking about the entire system, if you upregulate this gene, you could expect that from pyruvate to acetyl-CoA you will get more flux, right? How could you guarantee that from acetyl-CoA you will get most of the flux through this direction? You cannot guarantee that, because from acetyl-CoA there are so many other byproducts. Do you see where I am going with all this? So, systems biology basically gives you the opportunity to look at this system from a systems perspective. So, we do not want to study a single pathway just as it itself. We would like to study the entire system. We would like to know if I make some changes on a given gene or a protein, what will be the effect on the system. And if you take that into account, then you will figure that this manipulation will not work. Is it convincing enough? Why we need to have a systems biology based approach? Any question? Yes.
So basically in metabolic engineering, if you take the systems level picture, then the whole system should be known. What I am trying to tell, if you have some organism, do we know all the pathway from where it is goes?
That is a very good question. The answer is no. We only know about these pathways for well characterized organisms such as yeast, E. coli, some of the cyanobacteria. To give you an example, E. coli is the mostly studied organism, and even for E. coli 35 to 40% genes have either hypothetical or unknown functions. So, we have to work on basically what we know. So, to give you the answer, for model organism, for well characterized organism, we have better ideas. For some like obscure organism, this knowledge would be limiting. Yes.
Metabolic pathways for model organism is known, but when I am working with this, I am working with one microbial still. So if I got to study the metabolic flux, means how the flux is shifting and how the pathways is, means what are the products is
Right.
being produced.
Mm-hmm.
So how can I relate with the model organism? Can it be possible to relate this?
So, if I understand your question correctly, you are asking, can we study this system in a model organism and take the learning and apply it in a non-model organism? Is that what you are asking? Sometimes it will be good, most of the times it will not be good.
Because that can produce another product
And also if you need to take into account the regulation and other things, that will be totally different. Although let us say we know about the phylogenetic tree, we know that if you are working in organism A, which is a very close neighbor to organism B, if you are doing that kind of stuff, chances of being successful will be higher than organism A and B, which are like way apart in the phylogenetic tree. Does that answer your question?
Yeah, but that is a limitation of.
That is a limitation, but which is why we'd like to apply a model driven approach, which I'll discuss. All right. So what I'm trying to get here is basically when we are engineering any microbe, you will find some challenges. One of the challenges is, do we know all these pathways? Like as we are discussing, do you know all these pathways? Can we build a metabolic network model? That is sometimes limiting. Even if you look in literature, you will not find many different models, many different good models. And what do I mean by good model? I will explain tomorrow, not today, but hold on to that thought. Another challenge that you will find is when you look about the synthetic part that you can assemble together to build a synthetic circuit, for instance, you will see that most of the times these parts are meant for model organism. You will not find many different parts that you can use like readily for a non-model organism. And another important aspect is, what about the other mechanism? Can we know about the regulation, signal transduction, and other things that happen in a non-model organism? So long story short, if you combine these three, these are the major challenges that you will face to engineer a microbe. This is a common theme. It may or may not happen for all different systems. Maybe for a system you have a metabolic model, but you will not have synthetic biology tools, or other way around. You will see a mix and match of all these three different challenges. So what will happen in the next few slides, I will basically talk about 4 different examples where people have either used a systems biology driven synthetic biology or just plain and simple synthetic biology based on some rationale. So here is the first example. How many of you have heard about Greg Stephanopoulos? Good. So Greg Stephanopoulos is called to be the father of metabolic engineering, and he has been working on this area for, I don't know, 25, 30 years. So one of the system that he works on is Yarrowia lipolytica. As I said, this is an yeast, but not the Baker's yeast that we know. It's not Saccharomyces cerevisiae, but a close relative. What it's known for? It's known for producing lipid fatty acids, which could be used for many different applications. So what he's trying to do, how can we convert carbohydrates to this fatty acid or lipid molecules? Previously, people have thought about some engineering techniques which haven't worked, because I know that one of you is a chemical engineer, but rest of you are not. So to give you a quick example, let's say you would like to upregulate a gene, which is the last gene of a metabolic pathway, last gene, and you are upregulating that gene. Do you think by upregulating that gene you can increase the product?
In between there may be a reversible pathway, or and the last product can be used in the other directions also.
Good. Anything else? The question is, you are upregulating a product and you are just you are increasing the product and you would like to upregulate the gene at the last step. The question is, do you think it will work all the time? Sorry? Good. So if I combine both of your answers, that is basically what I'm looking for. Whether the upregulation will work or not, that will depend whether that step is the rate limiting step. If that's the bottleneck, then maybe it will work. If that's not the bottleneck, it will not work, because the bottleneck might be upstream. So that's what this work is trying to address. There are other mechanisms. As he has mentioned, your final product could be used as a precursor to produce other products. Your final product could be part of a regulatory mechanism, a feedback loop. There could be many different things. So when you are engineering, you have to take all this into account. So that's what they are trying to do. So what they are trying to do, basically they call it a push and pull mechanism. So what you would like to do, if you would like to increase the product of a specific product, you would like to increase the flow upstream. So basically you are pushing more flux towards your product, and you are also upregulating some step downstream. So you are pulling more flux. So that's a very well known mechanism in metabolic engineering, which will not need any metabolic model built. Just so that you know, this is something that they have applied without using a wholesale systems biology approach. This is purely based on their knowledge on the system and some rationale. So let's discuss about it. So basically, let's say triacylglyceride. That's the molecule you would like to upregulate, you would like to maximize. You would like to maximize the product of this molecule. So as I said, if you don't consider any other step, so basically what's happening here, if I have to walk you through it. So glucose, this is the central carbon metabolism, TCA cycle. Since this is a eukaryotic organism, TCA cycle is inside mitochondria. So from the TCA cycle you will get citrate, which will be turned into acetyl CoA, and finally acyl CoA, and in endoplasmic reticulum you will get these lipid bodies or fatty acid bodies. So again, we would like to maximize triacylglyceride. If you just maximize or upregulate this gene, as we have discussed, you may or may not be able to increase the production level. What they are trying to say is that if you upregulate this gene here, then you are pushing more flux in this direction, and then you are upregulating this gene. So from this branch point, think about this branch point, you are pulling more flux to this direction. So just hold on to this thought. Like, let's do a thought experiment. Let's think about upregulating this gene, ACC. If you upregulate this gene, that means there is more flux towards this direction. It could go either this way or it could go this way, right? But if you upregulate this gene, that means that whenever the flux reaches here, more flux will be pulled towards this direction. That's why it's called the push and pull mechanism. And to give you the idea how they are successful, when they are upregulating just ACC, they are able to increase the productivity level almost twice. When they are upregulating this, they did it by a factor of 4, but when they are combining these two, they have increased the production level by 5 times. So this is an example when just by looking at the pathway of your interest, you can design an experiment based on some rationale. Now comes the question, why this gene? Why not this gene? Why we think about, you know, upregulate this gene versus that gene? That is the answer that, as not being an expert on this area, I don't know. I don't know how they have decided this specific gene, not this gene, or this gene but not this gene. That is something comes from your expert knowledge. So when I talk about systems biology driven metabolic engineering, that basically takes this whole experience thing out of the picture. You don't have to be a real expert to come up with this kind of strategies, because you will build a model, your model will give you some ideas like what are the genes you need to manipulate. You need to have an expert knowledge, which is required for this example. So to quickly recap what we have discussed the productivity of a specific molecule in yeast, one of the yeast strain, and the example is based on some rationale which is based on experience working in the system. But when you think about the rationale, this makes sense, right? Any question? Any question on the metabolic engineering approach for this specific example? Or has anyone, have you designed anything on yeast? Like, did you engineer yeast to produce something?
Not till now.
Not till now. But you could see some of the things that you could learn and then take in yeast, right? All right. So, the next example, it's from my doctoral lab. So, here I would like to give you an example of how you can use metabolic engineering based on a model driven predictions. So, this work came out almost 6-7 years back, I believe, and it's in one of the workhorse organism, E. coli. Although this is a photosynthetic microbe class, I would like to give you some ideas how people are working in other microbes, and the next two examples will be on photosynthetic microbe. So, E. coli, what are the benefits working in E. coli? Can anyone tell me? What are the benefits? Fast growth rate, easy to manipulate,
and the whole genome is characterized.
whole genome is characterized. I think you nailed it. So, anything else? Yeah. So, E. coli is basically the most well characterized organism, the most studied organism, like, and as I said, other than that unknown or hypothetical proteins or genes, we know a lot about E. coli. Yet, when we think about using E. coli to produce fatty acids, people have struggled to increase the production level of fatty acids. How many of you have known about the fatty acid production pathways or beta oxidation pathways? Can you like, what is a beta oxidation pathway? What it basically does?
The reversal of fatty acid. Like in a fatty acid, the malonyl CoA is added to the
Right.
acetyl CoA, and like over loops,
Yeah.
it is added like chain of 2.
Right.
It increases, and in beta oxidation,
It's basically the reverse.
peroxisomes, and it is broken down by chain of.
Exactly, it is. So, to recap, you are talking about fatty acid pathways of different chain length, 12, 14, 16, 18, and beta oxidation pathway is basically breaking down those chain. So, let us say if I give you a task, I asked you, could you maximize the production of C16 fatty acid or C18? So, you can definitely think about two different approaches, like I would like to maximize or upregulate the gene, maybe the last gene of the pathway which produces C16, right? Or you might think about, maybe I will downregulate the genes which are involved in degrading C16, right? So, you will see that if you take this kind of approach, it does not always work, and I will explain. So, for this work, they have used IF1260 model, but at that point that was the latest model. Now, we have two other models of E. coli which are probably two times larger than IF1260 model. So, they have used this model, and they have used an approach called opt force. Have anyone heard about opt force or opt strain? So, if you haven't, I will not give you a very detailed explanation, but what it basically does, it uses the model, it gives you some predictions. So, it will tell you that what are the genetic manipulations that you need to make to reach to your desired product level. But then again, you have to go and test those predictions in an experimental setting. You cannot trust this model results as they are. They are only predictions. Some of them will be good, some of them will not be good, because when we talk about metabolic model, they really do not take into account the regulation of signal transduction as they are. In metabolic model, we have a way to incorporate this information as an indirect information. We cannot directly incorporate everything in a metabolic model. So, what happened? They have used this approach opt force on a metabolic model, and they have come up with all these different strain engineering techniques. So, if you see a delta in this, in any of this, that basically means gene deletion. If you see some green circles, basically that means upregulation. If it is a red circle, that red box basically, it basically means downregulation. So, what it basically means, if I have to interpret, let us say this is mutant number 2, it basically says that if I delete either any of these 4 genes, I will be able to get this amount of C16. Here we are only talking about C16, and likewise you can interpret other results. And all these percentages, this is basically a percentage of theoretical maximum. the chemical engineering folk? What is the theoretical maximum in a given reaction? How do you so what is the yield in a reaction? How do you define yield? Sorry? Right. Do you want to add anything? Yeah, but what I'm asking, like, if I have to calculate the yield in a reaction, how do you do that? You are thinking too many steps ahead, but yes, you are absolutely right. So when we talk about yield, that basically says what's the maximum possible. So let's say as a microorganism, we all know about cell cycle, right? So we know about the stationary phase, exponential growth phase, we know the death phase. So what is the objective of the microorganism in the exponential growth phase? What is its objective? To grow, right? Stationary phase, no one knows. We can guess that there are some other objectives, but definitely growth should not be the only objective, otherwise it would have grown, right? So this percentage is basically a theoretical maximum, which basically means if we want to calculate what is the maximum possible amount of C16 that we can produce in a metabolic system, and it does not take into account growth. You can always ask me the question, how can a microbe survive if it does not grow, right? It has to survive, it has to grow before it produces something. But here again, as I said, this is the theoretical max. So hypothetically, let us assume that the microbe does not want to grow, it only wants to produce C16. What is the maximum it can produce? This percentage is basically a percentage of that amount. So back in your mind, you might think it does not really sound that high because it is only 22.57%. But think about it, it is 22.57 of what is maximum possible if we do not consider even growth. Why that is the case? Because if we consider growth and production of a specific product, you have to split what are the resources you have. You are taking sugar from outside to make energy and few other metabolites. If you have to spend a single amount of glucose to produce two different things, obviously you are splitting your resources. What I am asking, what is theoretically possible at the max? So that is what this amount is. So what they have done, they have just picked few mutants as suggested by the model, and they have tried to test them. So I know that it is a busy slide, but to give you an idea, so C16 is here, right? As I said, if I give you the task to increase the C16, you might want to say, I would like to upregulate this gene, plain and simple. What they have seen that by upregulating this gene does not work. It might increase the productivity, but it increases only by a slight amount. What they have figured that if they upregulate this gene and downregulate this gene, again this is the TCA cycle. If you look at this metabolic path, basically pyruvate produces acetyl-CoA, malonyl-CoA, and then it enters the chain elongation of fatty acid biosynthesis, which is basically the production of different length of fatty acids. Beta oxidation pathway is the opposite direction. You are degrading all these different chains. So back in your mind, you might think that if I could combine upregulation of this gene, which produces C16, and I would like to downregulate the gene which basically breaks C16, and then I would also like to downregulate this gene. So what is basically happening, if you downregulate this gene, less flux will go towards TCA, it will go more towards this path. It will produce your product, but then it will not enter the beta oxidation pathway as it was used to in the previous case. So you are combining three different interventions, upregulation, two downregulations, and if you combine all this, then you could see that this is these three different strain designs after 24 and 48 hours, and you could see for the last case, which combines all these three different cases, your productivity level is quite higher than what you have started with. So, this is a very good example about how models could help you to come up with strain design strategies which you will otherwise will not find. So, again to quickly recap, the first example was purely based on intuition or some logic. The second example is based on model driven approach and how that could be useful. One thing I would like you to remember, the model has recommended 8 different mutants, for instance. They tested all 8 of them, only 3 was successful. Just to give you an idea, don't live in the illusion that yeah, model is all good and every time you, whenever you will try to design, you will get like all those results that will be good. Many times it may not be good, but the take home message here is it may not be good, but definitely you will get non-intuitive solution. These kind of solution you cannot get purely by intuition. So, that is why we need to build good model, that is why we need good metabolic engineering strategies. Okay. So, I will stop and then I will say, now is there any question that you have on these 2 different examples? The next 2 examples are based on photosynthetic microbes, but so far I have discussed 2 examples in 2 different systems which are not photosynthetic, but I have discussed 2 different approaches. Any question?
So if the organism is not well characterized, how can we use this? I mean, like the pathways will not be known, or the model, like it will have to predict on the limited information.
Right. So, okay, just to repeat the question was, if an organism is not well characterized, then how can we develop a good model? The answer is it would be really challenging, and that is one of the project I am working on. So, there are ways you can try to, like, to come up with some strategies, borrowing information from related organism, that is one of the strategies. But long story short, you cannot get good predictions from a bad model, that is a given, that will not happen. Well, if it happens, then it happens by accident, you cannot take it for any other cases. Does that answer your question? But we will discuss about how to address this challenge. Yes.
So if my organism has a very close relative which is well characterized, so can I borrow information from that and use it for my organism?
Yeah, you can. Again, that would be your best bet given the knowledge base. We will only build model based on what we know. We cannot build anything based on what we don't know. So, if you have a closely related organism which is well characterized, yes, then you have to make use of that information. All right.
One question.
Yeah.
Of course, has been applied in E. coli successfully.
Yes.
Whether it is been applied to Saccharomyces cerevisiae?
Saccharomyces cerevisiae, I am not sure, but I applied for cyanobacteria, which I will discuss.
Okay, great.
Yes.
So, in this 8 models,
Yeah.
like while suggesting this 8 models, weren't that also an expertise? Like, didn't that needed an expertise for just building that 8 models out of so many possible models?
So, the answer is yes and no. Why yes? Because yes, let us say you do not have any experience in models. It might take some time to get used to what are the techniques that you might have to use. But then again, I will explain, I will discuss about what are the tools that you have. So, the work that I have discussed, when it was done in 2010, that platform was not there. It is called K-base, knowledge base. K-base is funded by DOE. It is meant for users like anyone who does not want to know details about mathematical modeling. Let us say I am an experimental biologist, or I am a synthetic biologist, I have a wet lab, I am working on an organism. What I need to know, what are the non-intuitive solutions. So, K-base will help you to come up with the predictions. So, that part now is well taken care of. Few years back, that was not the case. Do you have another question? Okay. All right, but you have a question, but I answered, right? Okay, all right. So,
actually, I tried, of course, in Saccharomyces cerevisiae.
Yeah.
I was not getting.
Again, I mean, as I said, it's possible, and so Oftforce application, based on my experience, worked well in microbe. yeast is not a microbe, it's a eukaryotic system. So, it is more complex, so it is possible. All right. So, the next example is in a photosynthetic microbe, which is called Synechococcus elongatus 7942, and it is done in James Liao's lab. Back in the days he was in UCLA, now he moved back to his native country. So, what they are trying to do is, again, photosynthetic microbe. What is the beauty of photosynthetic microbe? It can fix carbon dioxide, it can use sunlight, and these are freely available stuffs, right. The idea is, can we make use of this freely available stuff and make something useful? If we can do that, if we can do that in higher amount, if we can do that in faster fashion, then it could be successful. One of the criticisms that you will hear from people is, what is the doubling time of E. coli? 20 minutes. What about a cyanobacteria or an algae? Few hours at least, the minimum, right. That is the the pushback that you will hear from people. Oh, this is all good, but it is too slow. Well, we cannot we cannot deny that this is a slow process. The point is, can we develop a process by using, let us say, a huge surface area and do something useful? That is the question. So far that has not been done. So, again, coming back to my original question, what they are trying to do, they are trying to use carbon dioxide to produce isobutyraldehyde, and isobutyraldehyde can be used to produce isobutanol, and isobutanol can be used as a biofuel. So, that is plain and simple, that is their approach, right. And again, this is not a model driven approach, this is purely based on rationale by looking at a specific pathway, which we will discuss. All right. So, let us look at this pathway. So, basically this is Calvin cycle, which is involved in carbon dioxide. Basically that is the pathway that consumes carbon dioxide in many different photosynthetic systems. There are other pathways that I will discuss in the next class, but basically then you will produce pyruvate, and at the end you will get this compound ketoisovalerate, and then you can produce isobutyraldehyde. The one issue they have faced is that this organism does not have this gene. You remember at the beginning of this class I told you, like, let us say you are interested about a product, what are the challenges that you will face? Here is a challenge. You are interested to produce something, that organism inherently does not produce that. So, as a synthetic biologist, what will you do? What will you do? Introduce a pathway. How can you do that?
Some precursor is already present, and we can convert that particular component into a different product by adding gene or enzyme
Right.
responsible for conversion.
Exactly. So, the answer is, you can knock in an exogenous gene from a different organism and make sure the product is there. So, in that context, you have to have few considerations. For instance, there might be 100 different organisms which have this gene. How do you pick? How can you pick which one to pick? Some of you are doing metabolic engineering, and maybe you have already done that. Do you know the answer?
Homologous genes and optimized codon optimized genes.
Exactly. So, you need to find out probably an organism which is closely related again, but have this functionality. So, there is a chance that if you borrow that gene from that organism and codon optimize, meaning that if you codon optimize, you will get more proteins, and probably more proteins mean that will act as a catalyst in that reaction to produce this product of your interest. So, that is the first thing they have done. So, they have borrowed this gene from a lactobacillus strain, I forgot the name, but you can, this is the paper that I mentioned, you could go and check it. One thing that I did not mention, the reason why I asked you to sign up for this, because I will share all the slides and everything, all the papers will be there. So, please sign up for that canvas. All right. So, they have borrowed this gene, and now they can produce the product of your interest. How many of you have heard about neutral sites? In a genome, bacteria or other system, we have neutral sites. What are these neutral sites? What do they do? Have you heard about neutral sites? So, we know that in a genome we have genes, and I think we do not have to discuss about genes, but between genes there are sections of the genome which does not do anything, at least we do not know about their specific function. So, some of these organisms, those sections could be targeted to incorporate a foreign synthetic element. Why we need that? Because for some organisms it works better. Let us say if you could incorporate a foreign part in the genome versus you have a shuttle vector, a plasmid, you designed it and you just have it there. What are the challenges you might face if you have a plasmid based system? What are the challenges you have? You have to maintain the plasmid. Could you explain for the class what do you mean by that?
The plasmid will have to have a marker or any resistance gene,
right,
and will always have to culture the organism with that dropout or gene or that drug so that the plasmid gets maintained.
Right.
If we don't do that, the plasmid will degrade.
Okay.
And those characters will be lost.
Have everyone heard that?
Basically like plasmid instability.
Yes, stability. That's it. If you are designing a plasmid, and if you are basically putting a foreign synthetic part, let us say gene, under a promoter inside a microorganism, you need to make sure, you need to have the selection pressure so that that plasmid stays there. Stability could be an issue. Compared to that, think about a system where you have a foreign part, it is already incorporated in the genome, then you do not have to do it, right. Can we all agree that that is a better system if possible? The challenge is, for a given system you need to know the neutral sites, you need to know where are these neutral sites. Again, coming back to my non-model organism, for the non-model organisms we do not know, we have to characterize them. Fortunately for this organism, they knew about this neutral site. So, what they have done, they have designed a plasmid, basically a suicide vector, and then they incorporated this gene in neutral site 1, plain and simple. Then they have figured that then they could produce isobutylaldehyde, but now they are producing only tiny bit. If you think about this pathway, as I have asked this question probably few times, if I asked you, now the system can produce this, what can you do? Some of you might say, I would like to upregulate this gene, some of you might say this gene or this gene, right. This is a linear pathway, although we do not know about the regulation. We do not know what kind of regulation we do have, but you might want to come up with all these different kind of upregulation. It may or may not work. What they have done, okay, first of all, how can we upregulate a gene? What are the different ways a gene can be upregulated? Promoter strength could be changed. What else? I need an answer from the back. People are awfully quiet at the back. How we can upregulate a gene? Some of you work, if I remember, some of you work in metabolic engineering. How do you upregulate something? We heard about promoter strength. Maybe you swap the promoter, you have a promoter with, you might have an inducible promoter, that means if you have the inducible element, your gene expression might be higher, that might be one of the ways. What would be the other ways? You could incorporate a second copy or third copy of the same gene, right. If you have that, that is logical, right. So, let us say this organism has one of these genes, and if you incorporate a second copy of the same gene, you might guess that that will upregulate that gene, right. What they have done, they have found out these 3 different genes from 2 different organisms which are known to be more efficient of expressing these genes. So, basically they have developed an operon, meaning that they have these 3 genes under the control of one promoter, and they have incorporated it in a second neutral sites. So now going back to my original problem, the bug didn't produce isobutyraldehyde. So they have incorporated a gene, but we didn't reach our production goal. What they have figured, they would like to upregulate all these genes. In one of these designs, they have all these three together with the control of a promoter. And if you look at this SA665, which has this gene, an upregulation of 2, 3, 4 and 5, that was the best based on their experience. So they were able to produce isobutanol for about 7 hours, and you could see from the optical density the cells were basically in their stationary phase, and yet they were producing isobutanol. So now when they compared this, some of you are working on algae based biodiesel, right? I remember. So that was considered as a benchmark. So compared to that, how is their production level for isobutyraldehyde? And if you have isobutyraldehyde, you can produce isobutanol. And if you look at these two, they are pretty close. At least at the isobutyraldehyde is more than the benchmark. So that was their selling point. So they have engineered a system which can produce isobutyraldehyde. From isobutyraldehyde, you can produce many different products. That is more than the benchmark, and usually algal biodiesel is considered as a benchmark in this area. Again, so far what we have discussed. One example, kind of intuition rational based. One example, model based. Again, this example, this is not a model based. This is purely based on intuition, their experience, and testing few things. Now you could guess where I am going with all this. The next would be obviously model based, and that is the last example of this lecture. Any question before I talk about it?
In the previous stage,
Yeah.
isobutyraldehyde is converted into isobutanol inside the organism, or?
Yeah, inside the organism, yes.
So, from that previous slide.
Yeah.
So this after KDC, this isobutyraldehyde, there is again step 1.
Again, steps which are not inherently in this bug.
So
That is also designed. I didn't talk about it, yes, but good catch. All right. So this is one of my works that I did when I was finishing up my postdoc in Himadri's lab. Again, my entire PhD is based on model building and analyzing the models. In my postdoc, basically I did all experiments. But this is an example where I have used models, oft force in particular, as we are discussing, to come up with a product which is called limonene. And limonene can be used for many different applications in pharmaceuticals and others. What we know that this is 6803. When you think about photosynthetic microbe, this is the E. coli of photosynthetic microbe. This is well characterized. We know a lot about this bug. So that is the plus point. What is the minus point? It grows really slow. The doubling time is probably 5.5 or 6 hours compared to, as you heard, 20-25 minutes in E. coli. So before I discuss this example, you need to know, what is the relative growth rate of this bug versus E. coli.
So what is the growth rate for the PCC7?
That is, I think, 2.5 or 3 hours, faster than this.
Okay.
Faster than this. All right. So like our previous example, this bug doesn't have limonene synthase gene. This is the pathway, and limonene synthase is missing. So now the reason why I picked this example, so that you could gather a common theme here. What happened in the last time? The last step was missing. What we did, or what they did, basically they went back, they looked in literature, they found out what are the genes that can test. They have found out an organism which is known to be an efficient producer of isobutyraldehyde. They picked that gene from there. When we are working on this specific system, limonene synthase, we have found that these are the two different systems from where we can basically borrow limonene synthase. Then again, we borrowed these two genes, they are codon optimized, and both of them are incorporated in a neutral site of this bug. As I said, this is a well characterized bug, and we know about all the neutral sites in this organism. When we cultured for, I think for 7 days, we found that one of these two genes works better, which is basically mentha spicata, this gene, and you could see that the production level is higher for this. So we stuck to that. So we started with 2 different, and then we narrowed down to one which is more efficient in producing limonene. So now we have a system, a metabolically engineered system which can produce limonene. Okay, this is probably the most important slide of this class as far as the importance of models. If you understand this slide well, then my job tomorrow would be very easy. All right. So the person who is a chemical engineer, could you look at this diagram? Does it ring a bell? You have seen this diagram in your process control, in your other classes, right? So what is it? It's a pipe flow system. It could be like, maybe you have some fluid, maybe it's water, maybe it's something else. So what I would like you to think about, the rest of you may not have taken chemical engineering, but you know about all this, right? This is not rocket science. So let's draw a parallel. Fluid flow is carbon flux. The pipes are reactions, the joints are metabolites. What we are doing here, we have carbon source. Since it's a photosynthetic microbes, we will only consider carbon dioxide. And biomass is basically representation of growth, right? So let's see. What we'd like to do, we'd like to identify the pathways or manipulation targets by using a model. This is a representation of a metabolic model. So you could see this is the biomass, this is your desired product, limonene for our case. There could be a byproduct. So what will happen? Let's say this is the biomass case. So you have carbon dioxide, you can have flow through this direction, and then you can have flow through this direction, and finally you will have biomass. So what's happening when you are having a flow from here through this direction and channeling it to this direction, some of the flow will come here and it will produce a byproduct. And again, I am not talking about any engineering. This is plain and simple. You have a microbe, you want it to grow, and that's what's happening in the exponential growth phase, because you just want the microbe to grow. So it's using the carbon source, carbon dioxide, to grow, and it might produce some byproduct, may or may not produce, but for this example we are assuming it's producing. Okay. Next step, let's say now just look at this diagram, and now what's happening. So let's say there is a closure here. So now you have a flow through this system, and then you are producing biomass, and now at the same time you are producing a desired product. Did you see what happened? This is what was happening before. Now I have a closure here, and the flow is switched. You are still producing biomass, but you are producing your desired product. So what do you need to do? You need to make some changes. You need to make sure that certain flow doesn't flow through a specific part of the system, so that you can channel flux and get the desired product, right? But in biological system, you cannot do that. You cannot literally take out a system and say that, well, this is what it is at the moment. You have to design valves. By valves I mean you might have to upregulate or downregulate genes, but here that's what it's represented here. So if I could generate a valve here, here and here, I could say that now what I would like to do, click, I would like to open this valve so that when CO2 comes here, it flows from here, it produces our desired product. At the same time, it produces biomass. I will close this valve, I will close this valve so that there is no byproduct. What metabolic model does, or metabolic model based approach such as OptForce does, it will give you the suggestions for these valves. It will tell you what are the valves that you need to design so that you can get your desired product. Does it make sense? Please let me know if you have any question, because this is, as I said, this is the most important slides if you want to understand model driven metabolic engineering. This is exactly in very plain sense, this is what model driven metabolic engineering does. So, if I ask you what is the deciding factor for the success of this kind of a system, you might guess that it depends how complex, how complete, how comprehensive this flow network is. Let us say, to answer your question, if you are dealing with a non-model organism, that means that your pi flow network will not be comprehensive. You are already missing out some of the pathways. So, maybe your model is saying something, that is why it does not make sense. That is why even for E. coli, the model has suggested 8 different manipulations, only 3 was successful, because even with all this information we are still missing out something. But if we are missing out something, that is still fine, as long as we are not missing out the most of the things. Make sense? All right. So, just to quickly recap, this is the pathway that I shown before. So, this is limonene, 2 different kinds of limonene. What people have done for limonene synthesis, they have upregulated some of the genes from this linear pathway with an expectation that that will increase the production level. It was not successful. What we have figured, that if I upregulate this gene or this gene, that means there will be more flux through this direction, and then if I upregulate this gene, now you could see a common theme, right? I am pushing more flux, I am pulling more flux, but this push and pull is not dependent on intuition, it is based on the predictions that the model made. Yes, you can sit, yeah, do not stand.
For this push and pull mechanism, means we are choosing some gene to upregulate.
Right.
So, is there any specific rule, or it's basically a trial and error method to choose this gene, means to upregulate, means whether this is giving the best result or
Got it. So, the question was whether this is a trial and error. Some sense it is, but most of the sense it is not. It is not trial and error in that sense that the model has given us few other strategies which we have tested. So, it has given us 4 different strategies, we tested all of them could increase the production level, but this one increased the most. At the end of the day, no matter what the model predicts, you have to go back, you have to design the corresponding plasmids, transfer the foreign element, test it, and make sure that the model predictions match. So, that part is kind of trial and error, because if you are not successful, you have to come back, refine the model, make another set of suggestions, and that is a closed loop approach. So, it is like you are building something, you are designing and building something, and then you are also refining the model. So, that is why it is a closed loop approach. But what happens if you see these bar graphs, when we have all these 3 genes, we could increase the production level. So, by plus means we are upregulating a specific gene. All right. Now, this is the last task for this lecture. So, let us say you are an engineer or a scientist and you are designing a bioprocessing plant, you have a photosynthetic microbe. What are the considerations that you have to think about if you would like to scale up? So, what you are doing in the lab, basically when we grow this microbe, we deal with what? 50 milliliters, right? 100 maybe. But when you are designing a plant, chemical plant or a bioprocessing facilities, that is not the scale we operate on. So, if you are scaling this up, what are the considerations that you can think of that you should consider? Yes. Mass transfer, okay. Mixing time, right. Shear force, anything else?
Some basic thing what is required, like if we are working in laboratory, so it is a closed system, but when we go for a open system, first thing would be contamination,
right,
and that is very tough task to convert this system into that system.
Right. Anything else? I heard something here. Would like to add something?
Sterilized environment kind of puts
Sterilized environment versus, yeah. I think, yeah, if I combine all of your answer, maybe you got them right. So obviously mass transfer limitations is one, contamination is one. One of the things that we always get in the US is the pushback about GMO. So when we are thinking about this genetically modified organism, we are thinking of this huge scale. Why? Because as I said, it's a slowly growing bug. You have to operate in huge scale to be successful. You need to make sure there is no adverse effect. You need to make sure that the organism that you have designed stays there and there itself. It doesn't go anywhere else and contaminate anything else. So these are the considerations that you need to think about. So I'll skip this part. We have discussed about it a little bit. So I'll end this lecture with a success story. It's not photosynthetic microbe again. So Genomatica is a company which is known to take up this kind of model driven approach and scale it up. Some of you might always have this question, okay, this is all good. Well, we know that this works in a lab environment, but what about practical application? So this is, I've just put together this example to convince you that it's not in the lab scale. If you put your resources, if you are following a systematic approach, you definitely can scale it up to an industrial level. So this is an example of producing butanediol by Genomatica, and they haven't revealed what kind of microorganism they are using, which is why we don't know. It's their trade secret, but I know for sure because one of the persons came and gave a talk in my university, and he talked about the usage of OptNoc, another tool that my doctoral lab developed. That is kind of the previous version of OptForce to get this done. So this is an example of a success story. To quickly recap what we have discussed so far, talked about bioproduction and what are the challenges that we can face. We have discussed 4 different examples. The first 2 are in yeast and E. coli. One of them is model based, the other one is purely intuition. Then we have talked about 2 examples in 2 different photosynthetic microbes. One is based on some intuition, the other one is a model based approach. And finally, I gave you one success story of bioproduction. So I'll stop here. Maybe we'll take a quick 5 minutes break.
5 minutes break.
And then come back and talk about what is the second lecture. So photosynthetic, I think most of them photosystems and other stuff. But anyway, let's take a 5 minutes break. Thank you.
Sir, doubts?
Yeah.
Sir, while modeling a metabolic pathway, sir, do you consider other kinds of network like protein-protein interaction network, gene regulatory network?
Very good question. So the question was, when we build a metabolic network model, do we consider protein-protein interaction network or gene regulatory network? The answer is yes, but the information is incorporated indirectly, and that's what we will discuss in the tutorial in the afternoon. But so if I forget, please remind me. So yeah, the answer is not directly but indirectly, yes. All right.
So we have one more lecture before going to lunch. After lunch, we
have reported that possibly this is how plant has acquired this photosynthetic capability. So cyanobacteria is somehow got transferred to an eukaryotic system by this, and from there you could see that different kind of system has evolved. One is chlorophyte or green algae, rhodophyte or red algae, or glaucophyte, which is another variation of this green algae, but it has a lipid layer around its cell membrane. So basically it has a cell wall, which is unlikely for a bacterial system. So that's the only difference. But long story short, even if it's a busy slide, what I need you to take away from this slide is how closely related a chloroplast is to a cyanobacteria. And my job is to convince you that this is what happened, and this is why cyanobacteria is important, because they are solely responsible to create this Earth's oxygenic environment, right? Okay, any question? Now many of you have studied electrochemistry and you know about this potential. So basically this diagram shows the evolution of this photosynthetic system, and I will walk you through this. So what's happening? So this BCHL, that is bacteriochlorophyll. So when they have evolved, basically they figured that there is a molecule which get reduced and it gives away its electron. And when you look at the second stage, it's the same compared to this. The only difference is you can now you can see a manganese catalyst. The manganese catalyst can oxidize this hydrogen peroxide to oxygen and give away electron. The only issue is this was not efficient. So yeah, it can do it, but the efficiency level is really slow. What happens in the next step? The manganese catalyst got attached, and now this chlorophyll has been evolved to chlorophyll D, another version of chlorophyll, and they have become a little bit more efficient. And finally, the final step, this whole manganese system got completely incorporated in chlorophyll, and now we are seeing here this oxygenogenic photosynthesis, which is very common in plant, in cyanobacteria. So from water to oxygen we get all the electrons and then it moves on. That's the electron transport chain in a photosynthetic system. So this is what you will see in a plant, you will see in a cyanobacteria. This is what you will see in purple bacteria even these days. Purple bacteria are usually anaxogenic. They don't use oxygen, they don't use this kind of mechanism. They can use sulfur or other things like they sometimes use they have used ferric ferrous, that kind of compounds. So the take home message from here is there are different forms of photosystems, and this is how basically the photosystems have been evolved into different systems. Now when we think about the potential, this is what it looks like. For this water oxygen system, it's about 0.7. For hydrogen peroxide and oxygen, it's about 0.4, and you could easily guess how it has become more efficient with the period of time. So this P680, 680 star, that basically shows the jump in photosystems 1 with an excitation of light, and this is the same jump for a purple bacterium system, and the difference is, so this is the basically the wavelength of the light, so which is 680 for photosystems 2 and 870 for the purple bacterium photosystems. Now for different bacterium, basically the way this thing operates is, let's say you are either using water carbon dioxide or you are using different compounds. What's happening here? Again, I know that it's a busy slide, probably too much information, especially for people who do not work in cyanobacteria or plant. But let's think about it. You are getting, if you are a cyanobacteria, you are getting your carbon source, which is carbon dioxide. You can fix carbon dioxide by Calvin-Benson cycle or any other mechanism, but you also need energy, because you need carbon dioxide, fine, you need energy from sunlight. Why do you need energy? Why even we are discussing all this? Can anyone give me an answer? I know that some of you work in plant system, some of you work in algae. Why when we are talking about the electron transport chain? Why it's important to study?
So to excite the electron, and that electron will pass. So sunlight is the one of the source to excite the.
Yes, we all agree with that, but let's say we know this process. Okay, sunlight comes, it excites electron pass from one to another, and yeah, it's all good. But why do you care? It's good, fine, but so what? One person at a time. Yes.
For 80% energy to the cell.
Exactly. Anyone else? Or the question has been answered? I think it is. All right. So, the point is we have to study this because this is the process photosynthetic cell makes ADP. Photosynthetic cells make NADPH. You need this to power all what's going on in the cell. The carbon dioxide fixing business by Calvin-Benson cycle, it's one of the most energy intensive process, because to fix one molecule of carbon dioxide you will need 9 molecule of ATP and 5 molecule of NADPH. Where will this come from? This is where it will come from. That is one example. The thing that I am trying to convince you all is there are many ways, many different photosynthetic bacteria do all these things. It is not one type. Photosynthetic system could be of many types. These are few examples. The common theme here is that you need to think about the wavelength that the corresponding photosystem is being operated on and what it is making. Is it just making ATP or it is making NADPH? So, this kind of scheme, let us focus on this because this is the most prevalent scheme that you will see in plant and cyano. This is known as the Z scheme. Why? I do not know, it is more like N. I do not know why people call it Z, but it is what it is. I am not arguing that, but this is what it is known as the Z scheme. So, what happens in this case, in photosystems 2, you have water which breaks down to oxygen, you get electron, you excite the photosystem, and then the electron will be passed on to different compounds, and then finally it will reach to photosystem 1. One other thing I do not understand, how come you start with photosystems 2? Why not that one is photosystem 1 and the other one is photosystems 2? The answer is probably the photosystem 1 is discovered first before photosystems 2, but so that's what I heard again. So, in photosystems 2 again you will get light, but it operates at 700 nanometer, it gets excited and then the electron will be passed on. From here some of the electrons will come back. So, you could see that this flow of electron, this is linear. It flows from here, goes there, and then it goes here and come back here. This flow of electron, this is known as the cyclic electron flow. The linear electron flow is responsible to produce ATP. The linear electron flow is responsible for both ATP and NADPH, whereas cyclic electron flow is responsible to produce ATP. I'll explain in the next slide. But this is what you will see. Now, corresponding scheme you can see in purple bacterium, it has just one photosystem, not two, just one, because it does not want to break down, it does not want to get electron from water, right. So, it does not need that part of the photosystem. So, it has one photosystem to all these things. For green sulfur bacterium, same concept, the only difference is this wavelength 870, 840 versus 680 and 700. Again, these are different schemes that different photosynthetic microbes operate on. That is the take home message. You do not have to take all this information, but I would like you to take this information and this information only.
Okay. So, all those schemes, I mean all those bacteria with these schemes, they fix carbon dioxide or?
Yes.
Okay.
Yeah.
So now my question is, like, when we are choosing cyanobacteria for the bioproduction,
yes.
Why is that? You can choose the other two?
Yes. Yes, you can, because as I said, I worked with cyanobacteria for my doctoral and postdoctoral work. Currently, I do not work with cyanobacteria. I work with the purple bacteria, which I will discuss. And I think rather than sticking to a system, we should always ask ourselves the question, what is the problem? So, the question should be from, like, it is kind of a backward design. We know the problem, we will choose the best organism that can solve the problem, and then we will design that organism. That should be the logical approach. Many times that is not the case. Why? Because let us say I am a professor and I have been working on this area on a specific bug for 10 years. I do not want to change most of the times. So, that is what it is. For me, I did want to change for two reasons. One, I do not want to compete with my doctoral, postdoctoral advisors. They have been working in this area for 20 plus years. I cannot compete with them. Second, I wanted to get a different interesting problem. The bug that I work on currently, that is a purple bacterium, which are this kind of a system, not this one. And the cool thing about that bug is it can fix carbon dioxide, obviously it can use sunlight, it can use lignin breakdown product, which is kind of huge in the US, because I live in Nebraska. I don't know how many of you know about Nebraska. I'll not be surprised if you don't know about Nebraska, because it's called a flyover state, but no one knows about what's happening in the Midwest. But anyway, so corn is the major product, agricultural product in Nebraska. So many times people get sugar from the cellulosic part of plant, and they'll leave all this lignin based material as a waste. My goal is to make something useful from this waste by using a bug which can also use carbon dioxide and sunlight. So, I am using waste product and something that is readily available, doesn't cost us anything, to produce something useful. So, yeah, you are absolutely right. Why do we have to even stick to cyano? No, we can stick to other systems that are more efficient. So, even if you don't understand what I explained in the past few slides, this is something I definitely want you to take away from this lecture, just so that you can understand what's the basic difference. So, if we think about oxygenic photosynthesis, so basically we are talking about carbon dioxide, water, light energy, and this is what basically happens. It produces glucose, oxygen and water. We all know that. I think we have learned about it in our high school biology. What we haven't learned, or we haven't really focused on, the anaxogenic photosynthesis. So, just to draw a parallel, in anaxogenic photosynthesis, you will not have water as electron donor, you will have something else. I said H2A, it could be water, most of the times it is not, but it could be H2S, hydrogen or other electron donor. The system that I do work on, that gathers all its electron from an organic substrate. It could be acetate or it could be monolignol, lignin breakdown product. But at the end, it produces a carbohydrate which may or may not be glucose, something other than glucose, maybe sucrose and water. So, these are the basic difference between oxygenic and anaxogenic photosynthesis. Is it clear? Any question?
Yeah, so this H2 and either it's H2S or H2 or any other electron donor.
Yeah.
Is it specific for specific bacteria?
Yes, specific for specific genus of bacteria, family of bacteria, yes. Which is why my bug is known as purple non-sulfur bacteria. Then there is also a type called purple sulfur bacteria. And for purple sulfur bacteria, you could guess the donor would be H2S, right? Okay. So, this is basically from my lab, and if you look at the color, it really looks cool. Like look at how purple. I don't know, it's not purple actually, it looked more pinkish to me if you ask me, but anyway. So, this is this bug, Rhodopseudomonas pluristis, that is the name of this bug, and it is a gram negative bacteria, and as I said, purple bacterium could be sulfur or non-sulfur. It could, so the good thing about this bug is it can operate in low light. It can operate in low light, it is found at the sediment of different water bodies. You could easily guess at the sediment at the lower level, you will not get much light, right. So, you need to evolve your system in such a way so that you can operate in that level. You can scavenge what's out there, other carbon sources, right. And then there are green bacterium which can be sulfur, non-sulfur, but they don't use Calvin cycle as other cyanos do. There are also no other gram positive bacteria, which is heliobacteria. So, all these are anoxygenic phototrophs, they do not operate with oxygen, right. Now, contrary to that, you could guess there are oxygenic phototrophs, cyanobacteria is one of them. It could be single cellular or it could be filamentous. It can fix nitrogen, it can be used to produce many different products, and there are algae and land plants. We have discussed about it and how this have borrowed the photosynthetic apparatus from cyanobacteria. Now, as I said, this thing probably I could skip. So, we have discussed about linear and cyclic electron flow. We have discussed how this cyclic electron flow could be used to produce ATP, whereas linear electron flow is needed to produce both ATP and NADPH. The reason why I would like to show you here is that this is something that most of the oxygenic phototrophs have. That is the theme you will see. Whereas this cyclic electron flow, this is used by some of the photosynthetic microbes, not all, and they have used different pigments. These pigments, they are not same, but similar in that system, in all these different systems. Okay. I will stop here, and this is the question that I would like to ask you. What mechanistic difference you might see for a bacteria or bacterial species that you can find in north or south pole versus equator? What are the changes you think? So, we have talked about oxygenic, anoxygenic photosynthesis. We have talked about different bacterial system which operates under different wavelength. So, let us say I have asked one group of you to go either north or south pole, and like you might have gotten some sample, and the other group have gone to some equatorial space. What would be your chances? The group which went to either north or south pole, what kind of photosynthetic microbe you will most likely get? What is the limiting factor there? Temperature, sunlight, and oxygen level as well, little bit less, whereas in equator lots of sunlight, that these are not a concern. So, could we guess like what kind of phototrophs we will get in equator? And what about north or south pole? And okay, and so I heard mostly equator, it could be like plain and simple oxygenic photosynthesis phototroph. In the equator, in the north or south pole, it could be the other ones which operate in low light, low oxygen, that kind of thing, right? So, it does make sense, right? Anything else?
I just question and
Yeah, no, please.
Other than the answer.
Yeah, no, ask question.
So, if you go to the poles, if there is a like a kind of a 6 months of the dark,
Yeah.
how the phototroph can take the energy from the light?
So, that is the beauty of the system that I work on. Many of the photosynthetic systems, they operate in different mode. For instance, the bug that I work with, it can operate in the heterotrophic system. It does not need carbon dioxide or light. It can just survive by taking succinate, to give you an example. So, that is how these bugs are fascinating, because they are so flexible. Given the environment, they can change. So, survival, they have to survive. And even for the workhorse 6803, it can also operate on phototrophic situation, heterotrophic situation, and mixotrophic situation. So, yeah, that's probably your answer. Okay. So, this is another slide. Again, it's a busy slide, I understand. So, bear with me. So, this is just to show you. So, now we have talked about electron transport chain of different cyanobacteria or other photosynthetic system. The corresponding question would be, okay, so we understand the electron transport chain is different. Fine. You might guess that might mean that maybe the carbon dioxide fixation mechanism is also different. It is not same in everything. Most of the times when we talk about carbon dioxide fixation in a photosynthetic system, you might definitely have heard about Calvin-Benson cycle, which is this one. So, where you can fix 3 carbon dioxide to produce glyceraldehyde, 3-phosphate glyceraldehyde basically. And as I said, in one turn of this Calvin-Benson cycle, so that you can produce ribulose 1,5-bisphosphate, which can use the enzyme Rubisco, which is the most prevalent enzyme in this world, it's an energy intensive process. In one turn you will need 9 ATP, 5 NADPH. That's why you need to have the electron transport chain to produce all this. And as I said, this is, you can find purple phototrophs, algae and cyanobacteria, and plants as well, definitely. Now, when we think about green non-sulfur phototroph, it uses a different system. This cycle is known as hydroxypropanoid cycle. So, it basically can fix not carbon dioxide, but bicarbonate. It can fix 3 bicarbonate to produce pyruvate, and through this process it will require, I think, 5 ATP and 5 NADPH. And then there is green sulfur phototroph. We all know about TCA cycle, right? TCA cycle or Krebs cycle, and we know that it's a cycle that produces energy. So, in green sulfur phototroph, TCA cycle operate in the opposite direction. If you remember your biology, all these TCA cycle enzymes, they are bidirectional. So, the reaction could go either this or the opposite direction. The beauty of this bug is it operates on the opposite direction. Through this process it fixes carbon dioxide, and it will require 2 ATP and 4 NADPH. So, the common theme that you could see for all these different systems is all of them are energy intensive. If you want to operate in any of these domains, you have to have energy, which is why you have to use the electron transport chain to produce it. So, you could see most of the times when carbon dioxide fixation occur, electron transport chain has to be functional. Are we good? Okay. Now, this is one other aspect that you will probably hear in many different cyanos. How many of you have heard about photosynthetic microbes fixing nitrogen? A few. So, what can you tell me about nitrogen fixing enzyme nitrogenase? What do you know about nitrogenase? Do you know why this is a tricky business, fixing nitrogen?
There has to be absence of oxygen.
Why?
Because nitrogenase
is very oxygen sensitive. So, it has to operate under microoxic or anoxic environment. One other challenge is it is also very energy intensive. Look at this. It will require 16 ATP. So, it's very energy intensive. But the good thing is, which of the microorganisms or photosynthetic organism that can do it, it's very useful in that sense. You don't have to provide nitrogen in your environment, in your medium, for instance. It can fix nitrogen from the atmosphere, right? It's out there. There is no cost in it. The caveat is you need to make sure that the microorganism can produce enough energy so that it can fix it. Now, I always find it fascinating that how different cyanobacteria do it differently. So, when I worked in my postdoc with Himadri, he's kind of a well-known figure in this cyanobacterial world. One of the organism he worked on is called Cyanothysi 51142. This is an organism which can fix nitrogen. There is another organism which is known as Anabaena variabilis or nostoc. You know about this. So, the difference between this is a unicellular organism, this is a filamentous organism. So, why I find it fascinating? Because in the unicellular organism, can you see what is the challenge to fix nitrogen? Because we have discussed and we have established that nitrogen fixation has to occur in the microoxic or anoxic environment. Cyanobacteria needs oxygen. It evolves oxygen when it does photosynthesis, operates in light, right? Then how it does it? If you are a unicellular organism, how can you do it? because you are producing oxygen, but then you would also like to fix nitrogen. How will that happen? Sorry? At night. No sunlight. Okay. Anything else? Compartmentalize. Anything else? So probably not. So these are the two techniques that I am showing here. In one, there is a temporal separation, meaning that in light you will do photosynthesis, you will store glycogen, carbon storage. At night you will use glycogen as a carbon source, you will do nitrogen fixation, you will produce cyanophycine as a nitrogen source. You at the daytime you will use cyanophycine as a nitrogen source. So same cell, unicellular organism, but temporal separation of different processes. That is what it means, light dark. Whereas in anabaina, it develops a filamentous structure. One is called the vegetative cell, the other one is called heterocyst. So what happens in the vegetative cell? It is all simple, as like as other cyanos. It will fix carbon dioxide, it will go through this same electron transport chain, it will produce oxygen, and then it will store the fixed carbon in some form of sugar or carbohydrate. It will pass it to the heterocyst, which has a well developed cell structure, so that it will not let any oxygen in, or if it does, it will be very, very low. It will use the sugar, break it down by oxidative pentose phosphate pathway to produce NADPH. It has a photosystem 1, not 2, because photosystem 2 basically is involved to produce electron from water, and we do not want to have it. So it does have just photosystem 1, it will have the electron, and then this is your nitrogen fixation. Basically you will have nitrogen and you will have ammonia as a product. So this is a very good example how cyanobacteria handles this separation of CO2 fixation, light usage versus nitrogen fixation. One does it by spatial separation, the other one does it by temporal separation. Are we good? Okay. I will probably go quickly because we have already discussed. In the light period, you will have your carbon dioxide, your electron transport chain, and then you will move to your dark period where you will use the stored carbon. And interestingly, when we use the stored carbon, the metabolism basically operates like E. coli. It will breaks down the sugar, and then there will be plain and simple glycolysis, TCA and everything else. At the end, you will store nitrogen with cyanophycine, which you will use in the light period, and that is what cyanothesis 51142 does. Okay. Now, another discussion point. Let us say you isolated a mysterious microbes from the transition zone in a nearby marsh. How do you determine if it is a photosynthetic, oxygenic and or diazotrophic? By diazotrophic, basically I means that which can take nitrogen and produce ammonia, let us say. These are the diazotrophic cyanobacteria. So look at the question, tell me how do you decide? You can think it as a set of experiment that you might have to do. It may not be just one experiment, because I have asked too many things here, right? But what are the things, if you are thinking about, what are the things that you will want to do?
If I culture it under different parameters, like in one case light, in another case if I provide carbon source, and in another case if I use oxygen source, then it could be decisive in that way.
Good. Anything? You are trying to say something?
Yeah, like different conditions, the same way.
Right. And do you want to add something to that? Okay. So basically you have to test the organism in different conditions. And please say it for the class, what are the different conditions could be?
It could be light, it could be the source of chemical, oxygen source or nitrogen carbon source.
Good. So basically, in other words, you have to do this set of experiments to see what kind of an organism it is. And then, so if it's, let's say, first of all, if it's a photosynthetic organism and if it operates only under light or only with carbon dioxide, then you have to do that kind of an experiment, right? And if it's a diazotrophic or oxygenic, then you, so basically when you operate this kind of experiment, you have to always think about a control and another condition, right? Then you can decide whether this is a, you are, whether you can prove your hypothesis or disprove your hypothesis. So this is just to pick your brain about like what are the steps that people usually follow. So the reason why I pose this question is one of the strains that Himadri's lab isolated before I left that lab, that was isolated from a stream near I think University of Austin. I don't know who has thought about it, like they have seen a stream, someone fancied a thought like how about we take this sample and isolate something, and then they isolated one strain which grows like on a cyanobacteria standard really fast. So the doubling time, I think it's about 1.5 or 2 hours, which is still slow compared to E. coli, but think about cyanobacteria where most of the organisms grow 5 or 6 hours. So these are the things that you have to think about. I know that some of you are interested about evolutionary biology. So these are sometimes you will end up with finding something which is just out there, probably right next to this building, who knows. So that's how you can, you know, get a system which is more efficient. But the downside is when you get this strain isolated, then it's your job to characterize it. Until and unless you do that, no one will be able to use it. Forget about engineering. You have to first study that system to understand, and then you can think about engineering. All right, I had a small clip here which I cannot play because I cannot use my computer here. But how many of you have seen Raceway Pond? I know that some of you. So it's a huge pond, right? So most of the times in the US, so I went to a plant, a power plant last summer in Orlando, Florida. So basically what they do, they use this raceway pond and they are using the carbon dioxide that usually comes from this power plant. They are using this algae to just fix the carbon dioxide. They are not doing anything else. So in that context, these are the problems that you will encounter. Again, this is not probably a very good example, at least not from a bioproduction perspective, but this is just to show you how in larger scale these kind of organisms are being used at the moment. So again, when we think about this kind of organisms, first thing you need to know, what are the natural products? One of the things that I told you, that the bug that I am interested in, that can inherently produce this biodegradable plastic precursor, and so many other things that you can, hydrogen is one of them. The Synechococcus 51142, one thing I forgot to mention, when it fixes nitrogen to produce ammonia, it produces hydrogen as a byproduct. So some of the works are also directed towards this direction. So these are the common products that you will see in the cyanobacteria or the photosynthetic microbe world. And then again, these are the reports that you will see people have used cyanobacteria or photosynthetic system in microbial fuel cell, some other biofuels, one we have discussed, and so many others. So at the end, you need to first decide what product, as I said, backward design, what product, what organism, and how can you engineer. These are the questions that you have to answer. Okay, I should have shown this slide in my previous lecture, but I should thank all my folks in my lab. So this is basically my lab, my folks who work with me, and they basically put together the slides for me. So I should acknowledge their contribution. So, with that I will stop here. I will let you think about it. I will let you think about all that we have discussed. You have a notebook in front of you, right? What I need you to do, think about what we have learned. Again, you do not have to write an essay. I just need 1 or 2 statements, and also you need to tell me what are the things that is still confusing to you. Write these 2 things down and then pass it to me. What I will do, I will try to address those concerns that you do have. So, what I am basically asking, write what we have learned, 1 or 2 statements, and then write what are the things that are confusing to you. Again, 1 or 2 statements. I do not want you to write an essay again. All right.